All right, welcome back. We're moving on to histology, so the study of tissues. A little biology recap. Cellular anatomy, cells are not all the same, but they share the same general structures for the most part. Organized into three main regions: nucleus in the middle, cytoplasm, everything surrounding the nucleus, and then our plasma membrane making up the very outside layer. Some examples of cells and their functions: connect body parts, erythrocytes. Maybe it's not the most obvious example, but blood is traveling through, it's connecting our cardiovascular system with all the cells of our body. They might line organs such as epithelial cells, epithelium meaning the outside layer. Movement, skeletal muscle, obvious one. Store nutrients, fat cells. Fight disease, macrophage. Gather information, nerve cells. Reproduction, ovum or eggs. Should know all this stuff, but I thought I'd just do a quick coverage. Histology, cells are specialized for particular functions, and a group of cells that have similar structure and function together form tissue. So histology, we're concerned with tissue, not individual cells generally. Four primarily types: so epithelium, protection, secretion, absorption, and structure, again found on the outside of tissues. Connective tissue, support and structure. Nervous tissue, communication and control. And muscle tissue, movement, both external and internal. Outside of tissue, we have extracellular matrix. Contains the cell glue between cells, such as desmosomes, proteins, and other molecules. So starting off with epithelial tissue, they're going to protect, secrete, absorb, and filter. They have a high degree of cellularity, which means that these cells are going to be packed in really tight. They fit closely together with very little extracellular matrix, so they don't need a lot of support outside of the cells themselves. They contain specialized contacts, so structure features that facilitate tight association between cells. If you look over, let me turn my pen on. So we have some simple columnar cells over here, and in between them we have this adhesive belt, which is going to stick it together, and we have this desmosome, which is also going to help us stick together. Gap junction and tight junction, facilitating communication between the cells. They might have microvilli up top. They might have other kind of extra parts, but the basic structure we see here. Not necessarily the shape. This is one of the shapes. So that cell, those desmosomes, adherens, those tight junctions, all this allows these cells to be so packed that we form a barrier. We have gaps in the barrier. Like if we're looking here, these villi is probably part of the digestive tract. So we maximize the surface area of these epithelial cells to the lumen, or the open area that they're exposed to allow nutrient absorption. There's going to be ways of getting those nutrients through. There's going to be ways of secreting stuff like these goblet cells, but they're basically forming a barrier. The tissue always has one apical surface, so it's exposed to a lumen, cavity, or external environment. If you think of our skin, external environment. Lumen means that's a lumen, and we have these epithelial cells surrounding the lumen. Or you can think of like a cavity such as the stomach or any of our hollow organs. They might have any of these modifications, microvilli like we see in the microvilli and villi like we see in the intestinal tract, cilia we're going to see in our trachea, kind of the hairs on the end, and glycocalyx, which is a sugar that coats the outside. Most of the epithelium is avascular. It gets a blood supply. It needs to get nutrients. It needs to get waste out, but we don't actually have, like here we see the capillary running below. All the transport between our blood supply and these cells are going to happen through diffusion, just some movement through whatever intercellular space we do have. It does have a basement membrane. I could have picked a better picture, I guess, but it'll be down here. It's just kind of connection to the cells surrounding the epithelia, providing connection and reinforcement, as well as defining boundaries. So when we talk about the classification of epithelium, we are looking at the shape and the number of cells. So starting with squamous epithelium, it's kind of flattened out, cuboidal. It's roughly as tall as it is wide, and then columnar, it's much taller than it is wide. The number of cell layers. So here we have simple squamous cells. We only have it's one layer thick. Stratified, we're going to have multiple layers, more than one layer. Obviously, different uses. If we have simple epithelium, it's easier for things to cross that membrane and travel down. So if you think of our stomach lining, or not our stomach, that's probably stratified, but our intestinal lining, or even better, our alveolar surfaces, where we need that gas exchange to occur very easily, it's going to have a much lower number of layers, simple, down to one. Stratified, where we need more protection, probably think of the esophagus or the stomach, where we have things moving through. Or think of our skin, where we need that protection on the apical surface. So here we have a lumen. It's surrounded by simple cuboidal epithelial cells. Remember that it's for cuboidal, it's about as tall as it is wide, and simple is just one layer. And our basement membrane surrounding that. This is, I should have looked at wherever I got these pictures from. This is probably going to be in the kidneys, I believe. Lumen, single layer cells. Lumen, single layer cells. If we look over here, could have probably found a better picture here as well. This is our basement membrane going underneath. And here it's kind of hard to see our cells. These are going to be columnar, and we see the cilia on the end. Whether or not it's simple or stratified, we probably need a little bit better picture. It's not always going to look like, say this is simple, it's not going to look perfect like that. There's going to be things kind of winding, and I'm trying to do that with an image. And remember, it's in three-dimensional space, so not only winding this way, but also winding out towards the camera, or out towards our viewpoint. I couldn't make a complete I think this is our trachea. I think this is going to be simple. It's just not really easy to see, but it could also be stratified. One more I should mention: pseudo-stratified will kind of transition between simple and stratified as well. Moving on to connective tissue. This is a much shorter one than the biochemistry one. So our connective tissue is providing support and structure. It's found everywhere in the body. It's our most abundant tissue. It's going to bind body tissues together, support the body, provide protection, insulate to maintain body temperature, transportation of other molecules. So examples we see there, our fibrous connective tissue is going to be supporting the body. Our red blood cells, our erythrocytes, are going to be transporting other molecules. Our cartilage, kind of that smooth layer between these two bones, providing protection. It's providing that smooth surface. Fibrous connective tissue, loose connective tissue. This loose connective tissue is going to be insulating as well as providing protection. I guess I don't have any. Well, I guess you could say these this fibrous connective tissue in this tendon connecting muscle to bone is going to be binding body tissues together. So I did find a picture with all the examples. There's variations in blood supply. So some connective tissue is highly vascular, some connective tissue is avascular, some is in between. You think about something like bone, your first guess might be avascular, but bone is highly vascular. We have all that yellow bone marrow, red bone marrow creating all our blood cells. We have a lot of blood going through our bones. If we fracture a bone, it's gonna bleed a lot. Cartilage, on the other hand, you think of in your ear auricle, in your nose, in your surrounding your sternum. That's going to be avascular. It doesn't need a blood supply. It's just kind of there. Composed largely of extracellular matrix. So more matrix we have, less flexibility, more strength. When we talk about the specific cells of the connective tissue, so a few prefixes get you a long way. So fibro, associated with fibrous tissues, contains collagen fibers. Osteo is referring to bone. Chondro to cartilage. Hemo to blood. Cyto to cells. So using those prefixes combined with these 3 suffixes, we can cover a lot of the connective tissue. Remember from Human Body System, almost everyone, we had that bone remodeling assignment where we talked about the osteoblasts kind of rebuilding the bone tissue, the osteocytes kind of maintaining it, and the osteoclasts breaking it down to be rebuilt by the blast. Muscle tissue providing movement. So we have 3 types. And remember at the beginning I said internal and external movement. So you can think of skeletal muscle as kind of external movement, smooth muscle, and cardiac muscle providing internal movement. When we talk about skeletal, it's voluntary control, cells attached to connective tissue, cells are striated, and they are multinucleated. So the striations that we're seeing in skeletal muscle are going to be these bands coming up. They're obvious, like when I think it's the next slide, we're going to see a microscope image of it. These striations are really obvious when you're looking at skeletal muscle. Cardiac muscle has it too. And you see in this image, it's just as obvious. But when we look on an actual slide, it's not quite as obvious. I don't know if I differentiated it. No, I guess I didn't. It should be cells are very striated, whereas here cells are striated. If you look close enough, you can see it, but it's not always super obvious. Smooth muscle. Oh, and they're multinucleated. So here, this is all one cell going across. It goes a long way. Nuclei here, nuclei here. Sometimes it's not really obvious that it's multinucleated, but we'll see some examples both here and in class. Smooth muscle, so it's involuntary. Think of our digestive tract, pushing that food down, peristalsis. So it surrounds hollow organs. It's attached to other smooth muscle cells instead of attached to connective tissue here. It's not striated at all, and it is mononucleated. So for each cell, only one nucleus. These cells are going to be a lot shorter than the other 2 as well. And finally, and I didn't mention they're involuntary. We can't will our stomach to digest faster. Cardiac is also involuntary. We can influence our heart to beat faster or harder through hormones, through different chemical signals that we can send, but we can't like consciously. If you say you can meditate and slow your heart rate, that's true, but that's not like we can't just slow our heart rate down on its own. So that's involuntary. Found in the heart. I don't know why that apostrophe is there. May bifurcate. So bifurcation of a cell, it's going to look like this. It's just kind of splitting. They're striated. Like I said, not as striated as the skeletal muscle. They can be mononucleated or binucleated. So these all look pretty mononucleated. They can also have 2 nuclei per cell. If you look hard enough, maybe you find one with 3. But the skeletal muscle tissue is going to have a lot of nuclei, where cardiac is going to have 1, 2, or very, very few. So, guesses on here. One more thing to point out, these intercalated discs that we see on cardiac muscles can also help discover what's what. So first thing, look for the obvious striations here, right? So skeletal. We can see each one of these is a cell. We have on this one we have 1, 2, 3, 4, 5 nuclei just visible right there. So skeletal is always the easiest one to find. Now between these 2, I did find some really good pictures that made it obvious the differences, but it's not always this obvious when you're looking at the actual microscope. So here we have the intercalated discs. We have 1 nuclei, 2 nuclei on that cell. Looks like 1 nuclei. We have a bifurcation right here. So we have this one coming, kind of spreading, dividing right there. So we know this is cardiac. And finally, that just leaves smooth muscle left. We can see that this muscle, if you can see the ends of the cells, it's probably going to be smooth muscle. So here this cell ends. It's going to be attached to other smooth muscle cells around it. I should have kept these lines going. But it's going to be attached to it, basically to other cells surrounding it. and mononucleated as well. Let's see how we did. Easy, right? We'll do this in class with the microscopes. It'll be a little harder. But if you got that, then you'll probably be okay. Finally, nervous tissue, sense and think. Found within our brain, spinal cord, nerves, retina. Carries electrical signals. Our primary cell types are our neurons and our neuroglia. Usually people just say glial cells. So our neurons are doing the heavy lifting. Our glial cells are kind of supporting those neurons. Neurons, usually when you damage them, almost always they're non-regenerative. So we're not gonna fix dead neurons. Whereas glial cells, they can regenerate. Thinking about that, if you think of a brain tumor, tumors in general are the result of hyperplasia. So what type of cells do you think brain tumors are usually composed of? Neurons or glial cells? And there we see the neuron, we see the glial cells surrounding it. It's going to be the glial cells, right? Because they can regenerate. So when we have hyperplasia, an increase in the number of cells, it's going to be coming from these glial cells. When we have tissue damage, it can be the result of disorder of structure or function, from disease. It can be from trauma, so physical or psychological injury, congenital defects, so something that was present at birth. I made this slide a few days ago and I forgot I put the tissue damage in there. So yeah, it can have those three causes. When we repair that tissue, and I know it seems like it kind of jumped to something else completely different, but we're talking about tissue, right? So we got to talk about damage and repair. There are a number of ways we have to repair this issue. If you're in my patho class, you've seen this picture before. If we regenerate, it's the replacement of the destroyed tissue by proliferation of the same type of cells. So our cells divide, fill in that gap. We don't lose our structure. We don't lose our function. This generally regeneration is the best way we have to heal, although not all tissue can actually do it. When we have fibrosis, we don't have replacement of that same tissue. We have basically connective tissue, scar tissue coming in and filling that gap. So here you see we have fibrosis. It may not be obvious on the surface. This might heal pretty nice, but underneath we're going to have that scar tissue that's there. It's going to be avascular. It's not going to have any function. You see these microcapillaries that were there are no longer there. The type of healing that we have, regeneration or fibrosis, once again, it depends on the type of tissue involved, because we're not going to regenerate neural tissue. We're not going to regenerate cardiac muscle. There's lots of tissues that we can't regenerate. But if we have an injury to our skin or our liver, we do have the chance to proliferate those new cells to regenerate the tissue with full function. But it's going to depend on the type and severity of the injury. So a small laceration, easy to heal. If you remember from patho, first intention healing coming in from the sides, it's going to heal really nice. Versus here, where we had this much deeper injury, much wider, instead of first intention coming from the sides, we have second intention healing where it heals from the bottom, and that's generally going to be result in much more scar tissue. So here's kind of a injury breakdown. So we have whatever our injury is. We're going to have our cellular and vascular response. If that tissue is persistently damaged, if we have persistent tissue damage, we have our inflammation, we have our immune system coming in and fighting off whatever's going on, and that's going to last a long time. We're going to generate that scar tissue. Chronic inflammatory diseases, cirrhosis, chronic pancreatitis, pulmonary fibrosis. Obviously, it's depending where that fibrosis is occurring, we're going to lead to these long-term problems. Versus here, we have our cellular and vascular response. Our stimulus is removed. It's an acute injury. versus a chronic condition. Depending on the severity, so we have parenchymal cell death, but our tissue framework remains intact. So there's going to be a superficial wound. There's going to be some inflammatory process, but nothing like that persistent response that we see over here. We're going to allow regeneration of that normal tissue. So liver, skin, lobar pneumonia in the lungs, we can heal that. So that was that way. If we have parenchymal cell death and we have a damaged tissue framework resulting from deep wounds, that healing is going to involve fibrosis, scar tissue formation, organization of exudate. That's all that, remember, all that pus and immune cells and everything that's in there. We're kind of talking about halfway between regeneration, full function, fibrosis, no function. Here's we're going to have a mixture. There's going to be some tissue that's regenerated, some scar tissue that forms. Deep excisional wounds, myocardial infarction. For MI, if it's a severe MI, we lost all that oxygen supply. That tissue is mostly going to die, but there is some healing that occurs. I think that's the last slide. Yep, that's it. Let me know if you have any questions.